So let's begin. Today is the second session of this eternal suffering, and today we will have a hands-on experience with the hi-fi package and all of the DN over DS tools that we discussed earlier. So here's a brief disclaimer that you should better be acquainted with the lecture that was conducted yesterday. You can check the presentation that I will send today after the seminar in case you've missed something. Overall, I guess if you just remember the ADHD friendly summary, that will do it just fine. So, talking about GPT policy, while usage of ChatGPT is not prohibited, I think that you shouldn't really rely on it because from time to time, I guess it provides rather questionable takes on the DN over DS tools usage because even in modern papers there are some questionable decisions being made, especially considering the exploratory analysis. And well, you should better rely on what we taught you. And first things first, we have to set up our environment because in this notebook we will use both the bash, basically a command line tools, the that's the hi-fi package for you, and also the custom Python packages such as like file or KRR, which we discussed earlier. This is the only possible solution for the PGLS problem in the Python. So let's run the first cell. Let's wait a little bit. It will basically set up our Conda environment. Overall, I hope that this seminar will be brief because there's not a lot to discuss, really, and like everything is, I guess, not really that complicated. All of the theoretical basics were discussed earlier, so I hope that you at least remember something. And now it is restarting the kernel. Now we can run the second cell, the third one, which will stop our environment and install the hi-fi package. Let's wait for it to install. So yeah, as we can see, it is installed. Now we can just check whether the hi-fi is installed indeed. And yes, we see that the version is the most modern one. It's really important to get the more recent updates for hi-fi because the previous versions lack some important options that we will use in our pipeline. Let's import some libraries that we will later use. And let's clone the GitHub repo, which contains all of the data that is necessary for today's work and also your homework. So now let's discuss what we actually have here in terms of the data. In this walk, we will compare 17 species from the Chiroptera mammalian order. Basically, those are bats. And what makes them interesting in the context of aging research is a great disparity in their lifespans. Remember, during the lecture, we've discussed the differences between Myotis brandi and Myotis vivesi. Basically, two species that come from the same order, from the same genus. It's really interesting that phylogenetically they're extremely close, yet their lifespans drastically differ. And it is really interesting for us to conduct such an exploratory analysis and figure out which genes might be subject to positive selection, which genes might have some adaptations that allow for such an interesting biological phenomenon of prolonged longevity. Here we have our 17 species. They're divided into the two suborders, Yinptera Chiroptera, which we can see here, and Younger Chiroptera, which we can see here, the bigger one, basically. And here's the Myotis genus. All of those bats are long-living ones. Sadly, we do not have an annotated genome for Myotis vivedi, so therefore it falls out of our analysis. Here we can see actually the timeline and how and when they were separated, but that's not important for this analysis. We can actually add longevity quotient values from an age database, a database that contains all of the information about longevity of species, especially mammalian ones. And there's also a separate study only on bats in terms of their LQs. So we can basically classify bats into the three categories: the short-living ones, average-living ones, and long-living ones. And it is obvious that, well, LQ of 2 is actually relatively high. It is like we observe the lifespan, the maximum possible lifespan, that is 2 times higher than the one that we expect for such species. And for all other mammals, that would be extremely cool. But for bats, it is relatively uninteresting. And here we can see that all of the meiotis fall within the long-living category. However, we have two different bats that are also long-living ones, but they are not coming from the meiotis genus. This is Desmodus rotundus and Rhinolopus ferrumiculum. The Desmodus rotundus also does not exhibit hibernation, for example. And overall, in terms of ecological traits, it is quite different. Therefore, it is interesting for us to find out adaptations that are shared by all of those bats, despite the fact that the ecological traits, once again, might differ a lot. And therefore, they cannot be the ultimate explanation between the prolonged longevity some mammalian species, despite some claims. So, we have to acknowledge the fact that 17 species is a relatively low number for the DN over DS analysis, and the rule of thumb is to use at least 30. However, in this case, we just do not have such many meiotis, such many Chiropterian genomes annotated. It's currently there are only 25, and not all of them have data about longevity quotient. Therefore, we cannot use them for such an analysis. We can predict actually their LQs by using our models that we will construct in the end of this session. But overall, I just don't think that it is really reliable. Therefore, we can conduct the forthcoming procedure in the most careful way that is currently possible. And therefore, we have to rely on the complex pipeline, which we will discuss right now. So, for this exploratory analysis, we will begin with busted minus C. As previously discussed, this is the most strict and robust method that actually tends to avoid false positive results as much as possible due to the fact that all of the technical issues are synced into the error syn class, an additional omega class. And then we will figure out at which branches did the positive selection occur, if, well, if it occurs within the gene. For this task, we will use upsrel, which is an adaptive branch side like model, and it will identify at which branches is the positive selection occur. And well, busted pH, I guess you can use it from time to time just to double check the busted minus E results. However, in this case, we will skip it just for the sake of time convenience. And overall, it was a great method because it fitted both models, like with 3 omega classes simultaneously on the background branches and on the foreground and compare them. But overall, you cannot introduce an error syn class within it, therefore it became obsolete compared to the busted minus C. And then in the end, we will obtain root to tip omega values, which will be later used for the PGLS. For the root to tip calculation, we will use Muse Gout 94, because just like we previously discussed during lecture, for this task, you should not really use something really that complicated, and Muse Gout 94 will work just fine. So, moving on, we have our data folder, CBA comp-H homework, and within this folder, we have basically data for 3 genes, ETIC4, kininogen 1, and SLAs 3. During our seminar, we will use only ETIC4, and other 2 genes will be used during your homework. So ETIC4 is an extracellular matrix regulator. And actually, all of those genes are associated with the extracellular matrix, which is basically a hot new thing within the aging community. And it is, I guess, going to be discussed a whole lot during this course. And like if you read papers about aging, it is a topic that is relatively popular. So let's begin with DN over DS, and let's start with busted minus C. Well, I have the predefined function for it, in which the command line the command line command is here, and we can see that, well, the main part is actually this thing, and you can change it to like whatever you want. But in this case, we want to run busted, therefore we use busted. For example, if we want to run meme, you just change it to the meme like this. And since meme has all of the parameters shared with the busted tool, you shouldn't even touch anything that is here. Apart from the error scene class, you should probably delete that. So let's bring back busted. As we can see, there is an error sync parameter within the busted comment, and it is the one that introduces an error sync class. So in the alignment parameter, we obviously use our MSA. In the tree, we use our tree in the NuWik format. It is the most convenient one to use with the DNORDS tools. And here we have the predefined set of branches. They are predefined in the tree. So if you open the NuWik file in any text editor, like the notebook or whatever, you will see that some of the branches are labeled as either foreground or background. I guess I can download it and show you. Here we can see the foreground. Here you can see the background branches. Basically, if you look at our tree, foreground branches are those that are associated with the long-living beds. For example, the branch that is leading to Myotis brandy from their common ancestor with Myotis lucifagus, and obviously the branch that connects Myotis lucifagus to their common ancestor, and also the common ancestor of all Myotises. Because in this case, we have only Myotises that are long-living ones. If we will introduce the Myotis vivesi, we will have to reconfigure our tree and label branches accordingly. So let's define our function and let's actually run it and see what we will obtain. So it runs roughly from 5 to 10 minutes, depending on which machine did the call-up gave you. And in case you won the call-up lottery, you will get the one with really cool CPU, because CPU is the bottleneck for such analysis. The Hi-Fi cannot use GPU and and its parallel computing power. Therefore, we have to rely on the power of our CPU. And on the ones that are not really that powerful, we obtain the values of 10 minutes or even more. And so while we are waiting, let me tell you about the amazing argument that I've heard from the aging researcher Piotr Litsky, who is quite famous. And if you remember, I was previously talking about the disposable soma theory. Basically, it states that the main limitation, or the trade-off between the reproduction and the longevity, is the fact that the energy resource is limited. Basically, food is limited for animals, sun energy is limited for plants, and etc. But Piotr Litsky argues that if you observe the population of lions and zebras, and if you actually genocide all lions, then zebras will reproduce extremely fast, basically exponentially to the moon, and therefore the food is not the limitation here. This claim is rather insane, and I really hope that he finds peace within himself and figures out his evolutionary theory, constructs it. It is something about the evolution of pathogens that are associated with aging. But overall, I guess you should always, when you're reading some papers about evolutionary theories of aging, you should always be critical of them, and always remember that they should explain the disparities between, for example, Myotis brandy and Myotis vivacia, or squirrels and rats in an adequate way. Because if you can't explain those differences by using those theories, then it probably means that those theories are kind of faulty. And while we're waiting, I also have to note that if you do not like those complicated functions, you can run it just like most of the bioinformatic tools in one single line that is quite long, or you can do it in a more user-friendly way, however you like. And also I have to acknowledge that the busted output consists of two things: the log that it shows in the command line and the JSON files that it saves to your hard drive. And well, the log is really I guess, user-friendly. It is really convenient to look at it, and therefore we have to also save it because the JSON files are like, how can I say it? They're really big, and sometimes it is just impossible to understand like what is where within it. Therefore, sometimes it is just more faster and more convenient to just look at the log. Therefore, we're also saving it. So soon the analysis will be done. And also, like, I have to acknowledge that for your homework, you can just copy those cells. Those are already cells with the code that allows to parse the JSON files without any grievances, and therefore, like, feel free to use whatever is written here in your homework. While we're waiting, well, are there any questions about the theoretical aspects of DN over DS analysis? Okay, that's really great. It seems like everyone understands everything.
Meanwhile, you can tell us why you choose this gene, what it does. I don't know.
So we are talking about this gene and its function. Once again, it is an extracellular matrix regulator, and from what I know, it like its expression was detected only in liver, and the like any disruptions within its function lead to some really bad diseases associated with it. And overall, this gene is really interesting from the perspective that it actually provides a really good PGLS plot, which is rather insane for a single gene to do. We will get to this part, you will understand why it was chosen. Also, like I can say that from time to time, especially if you're looking at some studies about hominids, like for example, chimpanzees, humans, maybe some even Neanderthals one, um, there is a McDonald-Crittman test. This is basically an upgrade of DN over DS that allows to conduct populationary studies. And overall, it also relies on the DN over DS metric, but it introduces the differences between like DN over DS within the population and DN over DS like between different species. Overall, like it is used only, I guess, in the neurobiology when we are actually trying to explore the differences between, once again, chimpanzees, like some hominids, and humans, and how their brains evolved. It's really fun. You can probably check out some studies or ask Professor Filip Haitovich about this method. I guess he really likes it. The CPU power. It seems like.
What step requiring most of time?
So,
Alignment or
so like, I guess the first bottleneck is truly an alignment because if you have a lot of species and you have to conduct an alignment using mass-C, which also accounts for the amino acid translation, it requires a lot of RAM and a lot of like overall computational power, and it takes a lot of time just to prepare the data. And the second bottleneck is busted, the gene-wide selection analysis, because like you have to analyze all of the genes, and only then you can filter out those that exhibit positive selection from those that do not. And therefore, like the first two steps are the most, I guess, egregious ones. And later down the line, when you will run Upsrel, Meme, like MG94, whatever you want, it will be significantly faster. And overall, like running Upsrel takes around like 2 or 3 minutes, and running MG94 takes around 1 or 2 minutes compared to the Bastard. And it seems that we've gotten a short end of the stick because it takes more than almost 8 minutes, basically, sadly. And I also have to acknowledge the fact that Right now, as we can see, the third argument is foreground. Therefore, we are running our analysis for the foreground branches, for the long living best, basically. And like in order to conduct the truly comprehensive analysis, you have to also run the same analysis for the background branches. And just for the sake of like not wasting a lot of time, we won't do it at the seminar. However, in your homework, please do it. Meanwhile, while we're waiting, I also have a trivia fact for you. For example, the bat Myotis lucifagos is phenotypically absolutely identical to the bat Aptesicus fuscus, and we can look at them. But this one is cooler, it lives longer. Yeah, looks beautiful. So it seems that our analysis has been conducted. Let's check the results. Это случайно. Let's look at the log file because it is more convenient to look at it, since in terms of verbosity, it's significantly better. So what we can see is that there is an error absorption class, an error sync one, and the DN or DS value within it is insane, almost like basically consider it an infinity. And within like a small proportion, less than 1%, we actually see that this class fits pretty well. And we see that the p-value is rather small, and overall the episodic diversifying positive selection definitely happened within this gene.
Why do we have two negative selection rows?
Well, basically they are collapsed into the one class. From time to time, actually, it can fit two different negative selection classes if it explains the data better than just a single one. It happens. But here we have actually a one class because though this one collapsed into like the same one. We see that the DN over DS values are practically identical within those two classes. And also it fits like constraint model onto the foreground branches. Well, basically the branches that we define in the analysis in our function. And we see that this model also provides these results. And instead of the positive selection, well, diversifying selection, here we have the class that corresponds to the neutral evolution. The dn over ds is 1. And overall, it seems like this model didn't fit pretty well compared to the one with 3 omega classes. And like for your convenience, you can just check the main statistics here, the p-value, the LRT statistics, likelihood statistic of unconstrained model, the model with 3 omega classes on the predefined branches, and the one that is with only 2 omega classes. And overall, it is safe to assume that this gene definitely experienced some events of positive selection. You can once again either inspect the log file or like suffer with JSON, like whatever you want. And also I have to acknowledge that I guess the most optimal way, the most accurate one, is to first fit just ordinary busted up or like busted minus like what whichever version you want, and obtain hyperparameters for models that allow like for the best fit, and then fit once again busted minus c to further optimize it. Like this approach was proposed to me by the Professor Sergey Pond, but just imagine running two sets of busted analysis for like more than 1,000 genes. It is just insane, and nobody does that. So I really don't want to run the analysis for background branches, so let's just move on to upsrel. Here is the function for upsrel. In upsrel, we do not have to define the branches. Basically, all of the branches are tested for the positive selection. And that's, well, the idea behind the method, that we want to identify at which individual branches, like could the positive selection occur. And let's run upsrel. In your homework, you can just use those functions, but change the files. It's totally fine. And once again, it can be used like using only a single line, but like whatever you prefer. It shouldn't basically take more than 2 minutes, but we'll see how it goes. I also want to state that if you, for some reason, despise Python and love only R, you can conduct basically all of those analysis in R. And like, because like the hi-fi is just a combination of bash commands, and then you can just use their end results within like the some R notebook, like whatever you prefer. Once again, like we've discussed, there is a Caper package that does pretty much all of the necessary stuff. We see that there are two branches experiencing positive selection. We can once again examine the log file, which will be convenient for us, like since we are working with a single gene. And we can see the output. Basically, it prints all of the individual branches and the amount of like omega classes that can be fit within it. But I believe this is the first fit, and the second one will be here, the most optimized one. And we see that most of the branches can be explained with actually one omega rate by this model. And in this column that is called max DNDS, we observe first the value that is like the maximum observed within this gene for this species, and the proportion of sites upon which it was fit, and also the amount of sites that tend to possibly contribute to the positive selection events. Those are calculated using the empirical bias factor, and like then the most reliable thing, there's also a likelihood ratio statistic and the p-value. Overall, the upsrel later corrects p-values using the FDR correction. For like the default setting is Holm-Bonferroni, and after correction we have only two branches that exhibit positive selection events: Myotis lucifagos and Myotis brandi. You can check with species um, like via their tags. For example, ML stands for Myotis lucifagos. Myotis brandi has a tag of MB. And in this case, sadly, we lose out the Rhinolopus ferromecunum, who is also a long-living bat, who comes from the different suborder, actually. It was really interesting that we obtain, actually, the maximum DN over DS ratio. But since it is close to infinity, basically, it's more than 1,000, and it fits only on a small proportion of sites, that could possibly be due to some technical issues because Abstrail lacks error sync class. You can parse the JSON files using this cell, and you can observe the main statistics, the p-value, like the branches that experience positive selection, the proportion of sites that tend to exhibit positive selection events, and yada, yada, yada. Overall, now we are ready to conduct MG94 to tip the NDS calculation. And like the first things first, actually the MG94 model, well, the raw fit of it on the data sadly is available only in the different GitHub repo that isn't just hi-fi, but it is called hi-fi analysis. Why was it done in such a way? To this very day, for me, it remains unclear. I do not understand like why, especially considering that the MG94 basically is used within the apps rail and all of the other methods. So it's pretty odd. However, now let's clone this GitHub repo with MG94. And we can also create the symbolic link, but for me it didn't work. And so like you can try, but I'm not sure about the results. Overall, like if you have like if the help for the MG94 is displayed, like we see here, then it means that everything was installed fine. So everything is good. Now let's define our function for this MG94 model. Here we have predefined type lineage because there are multiple types of how this model can fit. There is a global type, which I believe averages like the whole gene out, but we are interested in the root to tip calculation. So we define the type as lineage. And let's run this command. You can once again run it in one line if you prefer. I believe it's one of the fastest running commands here, so it shouldn't take long. Especially considering that we have only 17 species, which is a relatively low number for such an analysis. So okay, it is done. Let's inspect the log. And here we can see a table where the first column is basically the name of the tip, which corresponds to like all of the species that we have. Second column is root to tip value. This is basically a branch length parameter, since the branch lengths are once again optimized by the hi-fi package further to like obtain the best possible fits. And then we have the root to tip dn over ds and also the confidence interval for it. But we will use just the dn over ds values. And we can see here that within the meiotis genus, the dn over ds values are relatively quite high. And the same can be said, for example, for the Rhinolopus ferrumecanum. But when we look at the Desmodus rotundus, it is an outlier within the long-living class. So let's move on. And let's create the CSV file that we will later use for the PGLS analysis and save into it all of the values that we've obtained. So within the JSON file, like there is the main part that called branch attributes. basically all of the information about each branch. And within it, the first key is the node name, and then we have a lot of parameters for it. And we are interested only in the parameter called lineage dn over ds. Well, basically the third column that we've seen in this log. It's this thing. Let's inspect how it looks. Everything is fine. And let's create a table with names of the species, full names, and their longevity quotients. Here's a dictionary that I've prepared for you with everything that you need. Let's run the cell. And now we have a data frame consisting of the gene names, well, basically tags, and then the NCBI IDs for them. Then we have the column that corresponds to the lineage DN over DS, the root to tip DN DS. Then we have species name and also longevity quotient for them. And now let's just check how much there is difference disparity between meiosis and other bets. As we can see, overall, the difference is immense. We can also actually show mean on the graph. So yeah, pretty much meiosis tend to exhibit significantly higher DN over DS values. Since this value, the root to tip one, is actually averaged across the gene, well, for each individual branch, it is significantly lower than 1. However, once again, via upscale and other means, we know that positive selection occurred for some proportion of the sites. So let's move on to the PGLS analysis. And for this, we have to actually save our CSV file, which is the one with the LQ name. And actually, just for the sake of convenience, restart the runtime, because there are some issues with Conda when we are trying to use it with some custom Python packages such as file_rr. Let's just restart it, just for the sake of convenience. And then we will just throw the file, the CSV one, into the co-op environment. I actually wanted to do it in a more deliberate way, in a more delicate way, but I haven't found the solution, and maybe I didn't try a lot. Who knows? Let's once again import all the necessary packages. Let's once again explore the data frame. So as we see, everything is good. However, there are some issues with the implementation of PGLS, and for fila_rr, we need a dedicated data frame that has only the values for independent variables and those for the target variables. So in this case, we should have a separate data frame consisting only of lineage DNDs and LQs. Therefore, we should drop all of the unnecessary columns and save it as separate data frame. Also, because it was done in a really, I guess, good practice, and overall the package is really well done and well thought out, we have to additionally save the list of species names. And also the order should be preserved. Once again, let's clone our GitHub repo with data. There you will find the special tree for PGLS, because in file_rr, there is a rather interesting issue. It actually fails to conduct the analysis if the internal nodes are labeled, because the auto thought that it just never happens. Nobody does label the internal nodes, so there are a lot of questions, and I know that they will never be answered. We can visualize the tree in just file a draw from the BioPython package, or we can do just an SCI art. You see that yes, this is our tree with the species names corresponding to the ones that we have in our species data frame, in our species txt. Also, I have to acknowledge that phyloKRR has an additional function of phylogenetically weighting the data. I don't know exactly how this works, and this is, from what I've seen, this is some hidden esoteric knowledge, and overall, it may be useful for the nonlinear kernels which are introduced in this package. But for PGLS, not only is it obsolete because it already does the phylogenetic weighting by introducing the phylogenetically associated matrix of variance covariance, well, basically the evolutionary distance matrix. Therefore, just in case you just want to fit the PGLS on your data, you should not use the weighting part, despite whatever like the tutorial tells you. As you can see, it basically does something similar to the phylogenetic independent contrasts. Overall, it is not a good idea to like fit the PGLS on this sort of data points. Let's actually do the PGLS regression, and we see that for a single gene, it produces relatively good results. What can I say? Let's look at the metrics, and R squared, like 0.42, almost 33, is relatively high, like for a single gene once again. I guess the main issue is the fact that this modus rotandus is an obvious outlier. It is a long-living bet that tends to fall within the left side of the plot, sadly. But overall, like we see that meiosis cluster pretty well, and also Rhinolopus is somewhere close with them. Overall, this is really interesting for a single gene once again. Like this pattern is a rare sight to behold. So now let's talk about your homework. The first task is to just reproduce the pipeline and additionally try to figure out at which codons did the positive selection occur. You can use the hi-fi meme for that. So once again, like I've told you, you can just copy one of the predefined functions and change either busted like or upsrel part to like hi-fi meme. And then just look at the overlap between the results. And also you can like, provide some biological interpretation. And I have to remind you that once again, all of those genes are associated with extracellular metrics. Therefore, they are quite interesting in terms of aging context. And also there is a small note, check the order of the species in all the data frames that you use for the PGLS. Basically use the order that is predefined here, like in our species txt. Then the second task is about building a multivariate PGLS model. Basically, you can use all of the genes omega values in order to conduct the more robust linear regression. And maybe you will obtain like significantly better results than either we observed for the ETIC4 gene. Or maybe it will actually be worse. Maybe there will be some gene that tends to bring some discrepancies with its omega values. You can also try to introduce a hibernation feature, the binary one. Like if beta hibernates, it is 1, if it doesn't, it is 0, obviously. And also I have to acknowledge that if you feel that phyloKRR is such a burden and you can't work with it, once again, you can use Caper in R. It is totally fine. Just provide the code here in the notebook. I'm not blind and I will differentiate the R code from the Python one. For hibernation data, you can use the bad data txt that is somewhere within our CBA comp H homework. It's here. Like it has actually a lot of ecological features also for bats, and you can explore it however you want, especially for the third task. And the third task is, I guess, a rather creative one. You should try to find the biological traits that actually correlate with LQs, or maybe absolute lifespan values, maybe a log transformed lifespan values, whatever you want. You can like use the papers that I provided you for reference. And like there is a database of animal traits that has body mass, metabolic rate, and brain size. You just have to find LQs. They can be found either using NH or like some other databases. Maybe you can opt to use just an absolute lifespan value that is also quite reasonable, I guess. Also, there is a rather interesting metric called innovation, and it is especially explored in birds. And you can try to find association between innovation and like lifespan of birds. Also, you have to account for the fact that for birds, the longevity quotient is calculated independently of other like non-flying mammals, basically. And there's also the first article here. It has a lot of traits discussed in the context of aging. Like, try whatever you want. And also, once again, in bad data, you can find things such as sexual dimorphism and k views and try to fit, like, for example, multivariate model containing hibernation, k views, sexual dimorphism, maybe like log body mass, whatever you want. And the main requirements is that you should have at least 15 species, and you should either explore, like, conduct a multi-clade analysis, basically try to find as many as possible, or you should primarily focus on the clades that are interesting in the aging from the aging perspective. For example, once again, here we've discussed Chiroptera order because it is really interesting, since there is a vast variety of lifespans. Or maybe you will explore just aves, birds, like whatever you want, maybe even amphibia. Or maybe once again you will conduct a multi-clade analysis where we will see comparisons between aves, mammals, and chiropterians. Like, well, chiropterians are part of mammals, but you got the idea. In terms of metrics, report their square and explain whether there is a strong association or not. If you remember the lecture, I said that the feature importances can be evaluated using Wald t-test. Sadly, in phyloKRR, it is not implemented because author was like, I don't know, he has his own views. He makes rather interesting choices. And I guess I can't really say bad things about him. Moving on, like the main thing that I want from you is like an adequate biological interpretation and the explanation of all of your choices, the biological implications of such correlation. Like, also, if you observe negative result, that is totally fine. Like, it happens from time to time. I perfectly understand. You have a week for that, and a week for such a task maybe isn't enough to like do it in a really, I guess, well thought out manner. But try your best. And once again, if you explain everything, like you explain your choices, you provide a good biological interpretation, and overall, like, it will be totally fine. You will get your points. So for today, that's pretty much it. You can begin now, or like, well, basically you're free to go. Do whatever you want.